is occurred if he mixed the heat killed smooth with the live rough and injected that group into the mouse died. And when he does an autopsy, he gets live type 3S. And so now that, because you know so much more than he did, what would you say caused the change in the type 3? Because type 3 was dead, type 2 was alive, mixed them together, injected into the mouse died. And then he does the autopsy, the type 3 are alive and well. So how do you explain that? How would you explain it? You all are scientists, biology. Give me a guess of what you think happened. Type 3 didn't die, will not work. You're a scientist, so you know it's dead. So let's say that this is true, face value, that they are dead. Like I say, bring your body temperature up to 109. There's no way you're gonna be walking around here. So it's not. That can't work. So no mistakes. So what is it? But see, the type 3 is dead. So if type 2 infects, it's still dead. That's better. Something from the dead cell went into the live cell and caused it to become a type 3. Does that make sense? Because Griffith had no idea of what it was that entered the live cell, but he said something entered it. And he's responsible for the term, the 1928 term, transformation, which basically states that something entered the live cell and caused it to be transformed. And we're not talking about cancer here. We're talking about going from a rough to a smooth. So obviously something entered. Now he didn't know what it was. It could have been DNA, it could have been RNA, could have been proteins, lipids. He didn't know what it was, but he did say something from the dead cell entered the live cells and transformed the live cells. And question.
Because the DNA on that, the one that causes the disease.
See, we don't know. I hadn't even heard of DNA. Now, if you say proteins, according to this, may have entered the other cells, or the polysaccharide went to the other cells. But of course, we know now the DNA from the dead cell went into the live cells and expressed itself. We know that now. But he could only say something from the dead cells entered it.
When you heat a cell up and when it becomes engaged, the DNA is still intact.
To 109 degrees will not cause it to be destroyed. Yeah.
So the organism itself would die, but the DNA would still.
Yeah. So when DNA becomes denatured, 212 degrees Fahrenheit. So 109 is a long way. And I hate doing this because, we're not supposed to talk in that term. We're supposed to say centigrade, 100 degrees centigrade, right? But I want to bring the point that we didn't heat it up enough to denature. So 109 degrees did not destroy the DNA. And so, it make me become verbose. When they say the virus is dead, I have a problem with that because virus is not living. You may have some agent that inactivates the virus. I like that better, but if you're going to kill the virus, you see that on Listerine and alcohols and gel to wash your hands and stuff. But you can't really do much to a virus other than destroy the protein coat. But we'll talk about that some more. But yeah, it doesn't for the most part, you didn't destroy the DNA because the organism is gone, right? So basically, the DNA is still viable. And that's what Griffith showed. So we attribute the transformation principle to Griffith. So right now, we may even call things transgenic. And what does that mean? You know, when you genetic engineering, that's all based here. Because when you put genes into a foreign organism, that's, you know, those genes are expressed. So that came much later. But at this level, we're talking about foreign genes entering another cell. So in 1954, I think, somewhere in there, Avery, McCarthy, McLeod, Avery, McCarthy, and McLeod proved that it was the DNA that caused the transformation. They did an experiment with RNA, DNA, proteins, lipids, and so on, and they proved that's what caused the transformation. In this experiment that's up here now, they're looking at that transforming principle, and I'm not going to go through it per se, but basically trying to illustrate how transformation can work and looking at it from another technique. I won't go through this experiment, but if you look at Avery, McCarthy, and McLeod, let me just ask one thing though. What do you think RNase does? RNase. What do you think it does? It destroys RNA. It's an enzyme that destroys RNA. What about DNase? It's an enzyme that destroys DNA. Now if you treat it with that, the DNA is gone or the RNA is gone. And so which one of those experiments would you think transformation is impossible? The top or the bottom? Yeah, because if you destroy the DNA, you can't get it. So I'm going to leave that there because we don't want to be here. This is another experiment looking at the bacteriophage. What is the bacteriophage? The phage, and this name is T2. Anybody remember what a bacteriophage is? It's a virus that attacks bacteria. That's all it attacks. So there are quite a few of them. So this is an example of a T phage, and you can see the head and the which is where the DNA is. Actually, all T viruses, T phages are DNA viruses. So you know you can have RNA viruses as well. Single-stranded RNA, double-stranded RNA. But in this particular kind of virus, they are double-stranded DNA viruses. And this particular virus attacks bacteria. And they are specific by the kind of bacteria it attacks. So here we'll be speaking of E. coli. So these will be the two life cycles. The lytic cycle, the virus injects its DNA. In the lytic cycle, the virus injects its DNA. This part, the protein part, doesn't go into the cell, just the DNA from the virus. And the virus goes in and replicates itself, and then make new virus, and then the virus, then the bacteria ruptures, and more virus will leave to attack other bacteria. They call that the lytic cycle. The virus goes in, replicates itself using the bacterial machinery. So all of the genes are the genes that the virus needs to replicate itself actually come from the bacteria. Hershey and Chase did another group of experiments that showed that the genetic material of virus, T2 virus, is DNA. So if I had to define a virus, I would say a virus